Hello dear friends and welcome. Today we shall talk about reproduction in algae. The main objectives of today's deliberation are: Number 1. To give basic concepts about reproduction processes in algae. Number 2. To gain knowledge about vegetative propagation in algae. Number 3. To understand asexual process of reproduction in algae. Number 4. To gain insight about sexual reproduction in algae. And finally, to make clear understanding about the conditions necessary for sexual reproduction in algae. To begin with, let me give a brief introduction. Algae are heterophotoautotrophic thalide organisms with either prokaryotic or eukaryotic cells, having an array of habit, structure, metabolism, and reproduction with a strong affinity to water. The cosmopolitan distribution of the algae at different geographic latitudes on all continents, covering entire biosphere, that is atmosphere, hydrosphere and lithosphere, is the result of wide adaptive variations. Algae are the most significant photosynthesizing organisms in the biosphere, as they capture more of the sun's energy and produce more oxygen than other plants. Algae support abundant animals as they create most of the foundation in aquatic food webs. They are of economic interest due to the presence of polysaccharides with thickening and gelling properties that are used at food and pharmaceutical level. Now, coming to the methods of reproduction in algae. Reproduction is a fundamental feature of all known life. Reproduction in algae can be vegetative, asexual or sexual. Vegetative reproduction occurs through fragmentation and many other ways. Asexual reproduction involves the formation of certain type of spores, whereas sexual reproduction takes place by fusion of two haploid gametes. Some species alternate between reproduction methods in succeeding generations. Some algal species can reproduce by more than one means depending upon the environmental conditions. Now let us discuss different types of algal reproduction. First is vegetative reproduction. It includes all those processes of propagation in which portions of the plant body become separated off to give rise to new individuals without any obvious changes in the protoplasts. It does not involve any spore formation and there is no alternation of generations. It's the most common method of reproduction in algae. It includes: Number 1. Cell division or fission. It's the simplest method of reproduction. The unicellular forms of algae commonly reproduce by this simple process, often called binary fission, as found in Chlamydomonas, Synechococcus, diatoms, etc. Here, mother cell divides mitotically into two daughter cells, which finally behave as new individuals. This is exclusive type of reproduction in Pleurococcus, some desmids, diatoms, etc. Number 2. Fragmentation. The plant body breaks into several parts or fragments, and each such fragment is capable of forming new thallus. This type of vegetative reproduction is commonly met within filamentous forms, for example, Eulothorax, Pyrogyra, Zygnema, etc. The fragmentation of colonies also takes place in several blue-green algae, for example, Aphnocapsa, Apnothis, nostoc, etc. The fragmentation may be accidental or by the formation of separation discs or by some other mechanical force or injury. Number 3. Hormogone formation. This type of vegetative reproduction is found in blue-green algae. The trichomes of blue-green algae break up within the sheath into many cell segments called hormogonia or hormogones. Hormogonia are commonly found in nostoc, Oscillatoria, Cylindrospermum, etc. Number 4. Hormospores or hormocysts. They are thick-walled hormones and produced in somewhat drier conditions. Now, adventitious branches. Adventitious branches are formed in large thyroid algae, which when detached from the plant body develop into new individuals. For example, fucus, dictyota. Now, budding. In protosiphon, bud-like structures are formed due to proliferation of vesicles delimited from the parental body. Now, secondary protonema. The thread-like vegetative bodies develop in the case of chara, which help in reproduction. Now, tubers. Tubers are spherical or globular bodies formed on lower nodes and rhizoids in chara. Tubers are formed due to storage of food. Now, bulbils. Tuber-like outgrowths are developed due to storage of food at the tip of rhizoids and on the lower nodes of chara called bulbils. After detachment from the plant body, bulbils grow into new plants. Now, amylum stars. Star-shaped aggregation of starch-containing cells develop on the lower nodes of chara. These structures are called amylum stars. When detached from the plant body, they grow into new plants. Now, echinates. The vegetative cells of certain filamentous algae develop into elongated, thick-walled, spore-like structures with abundant food reserves called echinates. For example, gliotrichia. They can tide over the unfavorable conditions. With the onset of favorable conditions, they germinate into new individuals. In most of the chlorophyceae members, the echinates are developed. Sometimes they are formed in chains. Each echinate may develop into a new plant, for example, Edogonium, Eulothox, etc. Now, coming to the second type of reproduction in algae, that is asexual reproduction. It's the process of rejuvenation of the protoplast without any sexual fusion. Usually the protoplast of a cell divides into several protoplasts and thereafter they escape from the mother and develop into new plants. Asexual reproduction occurs by the formation of spores. Depending upon the algal species, the spores can be produced in normal or specialized cells. They are either motile or non-motile. Each and every spore germinates into a new plant. In this method, there is no alternation of generations. Different types of spores are as: number 1, zoospores. The zoospores are flagellated asexual structures formed in zoosporangium. There may be single zoospore, for example Edogonium, or many zoospores, for example Cladophora, per zoosporangium. The zoospores can be biflagellate, for example Chlamydomonas, biflagellate and quadriflagellate, for example Eulothox, Cladophora, multiflagellate, for example Edogonium. In Vaucheria, a compound zoospore called synzoospore is formed. Each zoospore has a chloroplast and an eye spot. The zoospores may be either haploid or diploid. Zoospores are normally formed under favorable conditions. The zoospores are liberated either by the disintegration of the sporangial wall or by the formation of an apical pore on the zoosporangium. After liberation, the zoospores swim for a while, then withdraw their flagella and cyst and ultimately germinate into new plants. Number 2, aplanospores. Aplanospores are non-motile spores. These spores are formed either singly or its protoplast may divide to form many aplonospores inside sporangium during unfavorable conditions, especially in drought. For example, Eulothrix microspora. The aplonospores may also be formed in certain algae of semi-aquatic habitat. When they appear identical to the parental cell but smaller in size, they are referred to as autospores. For example, Scenedesmus, Colorilla, etc. Aplonospores with thickened wall and abundant food reserves are known as hypnospores and develop only in adverse conditions. For example, Pediastram, Sphaerula, etc. They are formed to overcome prolonged period of desiccation. With the onset of favorable conditions, the hypnospores either directly germinate into a new individual or their protoplasts may form zoospores. Due to deposition of hematochrome pigment in their walls, the hypnospores of Chlamydomonas nivales are red in color. Number 3. Exospores. In some algae, spores are regularly cut off at the exposed distal end of the protoplast in basipetal succession, called exospores. These spores aggregate in groups and develop new colonies. For example, Chamaecyphon. Number 4. Endospores. These are small spores formed within the cells. On the approach of favorable conditions, each endospore develops in a new individual. They are also called gonidia or gonidea. They are set free after the dissolution of mother wall. Without taking rest, the spores germinate directly and develop into a new plant. These are formed in many blue-green algae and Basidiophyceae. Number 5. Tetraspores. Diploid plants of some algae, for example Polysiphonia, produce a special type of haploid aplonospores called tetraspores, formed within tetrasporangium. The diploid nucleus of a tetrasporangium divides meiotically to form 4 haploid nuclei, which with little amount of protoplasm are developed into 4 tetraspores. After liberation, the tetraspores germinate to form male and female gametophytes. Tetraspores are non-motile spores formed in some members of Rhodophyceae and Phaeophyceae. In Polysiphonia, tetraspores are formed in tetrasporangia by reduction division on special tetrasporophytic plants. Number 6. Hypnospores. Hypnospores are thick-walled structures. These are formed during unfavorable conditions. Under prolonged unfavorable conditions, the protoplasm of hypnospores divides to make cysts. The cysts are capable of forming new thallus, for example, Chlamydomonas nivales. Number 7. Oxospores. In many members of Basidiophyceae, oxospores are produced. Each develops in a new plant. Number 8. Carpospores. In red algae, carposporangia are formed at the tip of gonymoblast filaments, which produce a single haploid or diploid carpospores. Each such spore develops in a new individual. 9th. Neutral spores. These spores are not formed within the sporangia. They are found in Rhodophyceae. 10th. Monospores. These spores develop within monosporangia. Each spore gives rise to a new plant. For example, many members of Rhodophyceae like Bangia, Porphyra, Porphyridium, etc. 11th. Paraspores. Such spores are reported from many members of Rhodophyceae. 12. Statospores. They are found in Xanthophyceae and Basidiophyceae, where they act as perinating bodies. 13. Gongresira stage of Vaucheria. In the aseptate filaments of Vaucheria, the protoplast divides into several parts. Several hypnospores or cysts are produced and the whole filament looks like an algal form, Gongresira. 14. Microspores. They are produced in many Basidiophyceae. Now, coming to the third type of algal reproduction, that is sexual reproduction. The occurrence of sexual reproduction is probably a mark of relatively advanced stage of evolution among the algae, since such reproduction is lacking or very rare in classes that possess a relatively low vegetative organization. For example, Xanthophyceae, Chrysophyceae, Mixophyceae. During sexual reproduction, gametes fuse to form zygote. The new genetic setup can develop by the fusion of gametes coming from the different parents. Conditions for sexual reproduction. The sexual reproduction takes place after considerable accumulation of food material and the climax of vegetative activity is over. The bright light is the major factor for the production of the gametes. A suitable pH value is required. The optimum temperature is necessary. Depending upon morphological and physiological characteristics of gametes, sexual reproduction can be of three main types, that is, autogamy, isogamy, and heterogamy. Autogamy. In autogamy, two gametes of same mother cell fuse to form diploid zygote. Since both gametes are formed by same cell, there is no genetic recombination. For example, diatoms. Isogamy. It is the process of union between two gametes which are morphologically and physiologically similar. After fusion, they form zygote. The gametes are called isogametes. Usually, they are flagellate. For example, Chlamydomonas, Eulothrix, Spirogyra, Zygnema, etc. In Chlamydomonas species, two vegetative cells may fuse to form a zygospore, that's hologamy. As a result of fusion of two gametes, the zygospore is formed. Heterogamy. The fusion of dissimilar gametes is called heterogamy. There are variations of it. Number 1, anisogamy. In anisogamy, the fusing gametes are morphologically as well as physiologically different. These are formed in different gametangia. The microgametes or male gametes are smaller, active, and formed in large number. The macrogametes or female gametes are larger, less active, and formed in relatively smaller number. When the two gametes are morphologically dissimilar, the anisogamy is said to be morphological. For example, Chlamydomonas bruni, Ectocarpus scandus. When the two gametes are morphologically similar but differ in their behavior, the anisogamy is said to be physiological. For example, Zygnema, Spirogyra, Ectocarpus siliculus, etc. In Spirogyra, the motile gamete may be called male and the stationary one female. Number 2, oogamy. It is the most advanced type of sexual reproduction. In this process, there is formation of unicell sex organs. The male sex organ is called antheridium and the female oogonium. The antheridium forms the male gametes or microgametes called antherozoites, which are generally flagellate. The oogonium forms a non-motile female gamete called egg. During fertilization, the antherozoites reach oogonium to fertilize egg and a diploid zygote is formed. For example, Chlamydomonas, Erogonium, Vaucheria, Cara, Laminaria, Sargassum, Polysiphonia, Batrachospermum, etc. In Erogonium, the Enthredia and Oogonia are formed in chains. A silk develops in the oogonial wall before fertilization and a receptive spot in the egg. In Sargassum, sex organs are formed in special pitcher-shaped depressions called conceptacles formed on receptacles. In red algae, Polysiphonia, the male gametes called spermatia are non-motile. The female sex organ, Carpogonium, is formed on a specialized filament. After fertilization, the zygote is transferred to another cell called auxiliary cell. The highest degree of specialization is seen in Cara, where the Enthredia and Oogonia appear to be surrounded by sterile cells. The structure so formed are called globule and nucleus respectively. As a result of fertilization, the zygote is formed, which secretes 1 to 2 thick walls and undergoes a period of rest. On the arrival of favorable conditions, it germinates. Usually, it undergoes meiosis to form meiospores. In some cases, it forms the diploid plants, for example, Cladophora ectocarpus. In Sargassum, the plants are diploid. Meiosis occurs at the time of gamete formation. Number 3. Aplonogamy or conjugation. It implies the fusion of two non-flagellate amoeboid gametes, aplonogametes. They are morphologically similar but physiologically dissimilar, for example, order conjugales. In freshwater algae, the sexual reproduction is best means of perennation because it is followed by the formation of thick-walled zygote or oospore. With this, we conclude today's lecture. Hope you have understood it well. Thank you.